agenda for today, but I thought we would cover a little bit about what we did in the kind of third day of the European Bioconductor meeting, where we kind of had a user developer day. I'm not sure we exactly knew who it was focused at, but just thought I'd sort of recap what happened because I guess about half the people stayed for that kind of day. And then I'm going to basically present the same kind of thing that I did at that meeting. Might be even quicker than 8 minutes with a slightly reduced slide deck, but I know there's a couple of people on this call that like HDF5 and want to know about advances in that kind of area and things who weren't at that meeting. And then we will see what we do for the final half hour or so. It may be I let everybody go early, but we'll see what happens. So we're on the final day. We basically had 4 breakout sessions, I guess, which were kind of self-organized. People volunteered topics. There was something on mass spectrometry, and I don't think Laurent has made it yet. If he pops up, he will summarize what they did in that session. There was a session on IC, one on multi-assay experiment, and one on experiment hub and how you might use that and things. So I guess I guess Charlotte, would you like to sort of cover what you were aiming to do, maybe what came out of the IC breakout session, what people thought about it?
Sure. So I can maybe share my screen. Does this work?
Yeah.
Yes.
Okay. So so we were, I think, 6 people in our little ICU breakout session. And maybe I thought I'd just give a couple of words on background, and then I'll show what we did. So for those of you who maybe don't know IC, this interactive exploration tool for summarized experiments objects. And if you kind of open it with the default settings or the simplest way, just typing IC of a single cell experiment object, you get something like this, right? So different types of panels that show the objects from different aspects. And maybe this is not what you want, right? So maybe you want something like this. Maybe you want just 3 different types of reduced dimension plots, or maybe something else. Maybe you don't care about the tables, or maybe you only want tables and so on. So you can kind of get to this in different ways. So either you do it interactively in the app, you can just move around panels and you can resize them and make new panels and remove panels. So that you would have to do like every time you run the app, you would have to do that interactively. You can also do it programmatically, so you can make the app start in this way, but basically you would have to kind of go to the settings in the app and export a couple of 100 lines of code that looks like this and basically sets every single element of the app. And then you can run this code and you can call IC again with these settings, and it will start exactly in the configuration you wanted. So what we wanted to do in our session was to kind of try to make this a little bit easier. So we would like to just have, at least for standard or kind of setups that many people would like to use, something where you could just do a single line of code again. So just something like, if you want to reduce dimension plots, you should just be able to do mode reduce dimension of your object, and it will open in this kind of configuration. So it would be as easy to call as if you would call it with the default settings. So that was the idea for some standard setups. Write this mode. So basically write these functions that sets up the app in a specific configuration so that it's easy to just start it in that way. So we kind of, these things we thought they shouldn't go directly into the basic IC package because they're not strictly necessary for running IC. So we set up this ICU or IC universe package. It's here on GitHub. And basically, if anyone would like to contribute, that would be great. You just go here, you can fork it and so on. So it's basically, we want to know how people would like to use IC and to make it easier for different applications. So what we did in the end, so Michael worked on exactly this reduced dimension mode. So this is actually now in the ICU package already. You can just call mode reduced dimension with an object, and it will make this setup where you have one panel per reduced dimension representation that you have in your object. And then you can also say, I want to color by this particular gene, and it will make this row statistics table where you can select genes. And if you select a gene, all these panels will be colored by the expression level of that gene. So it's a kind of a simple way to easily compare different dimension reductions of the same data. So that's kind of the one that I guess went furthest and actually made it into the package. The other things are still kind of work in progress. So Federico worked on this differential expression mode for bulk RNA-seq data. So here you would call it again with a single cell experiment object because you want to be able to store like an MDS plot or so. So this shows MDS plot of the samples. Here you have like the expression level of a given gene that you can select in this table here. You have an MA plot, and you have like a volcano plot, and then another gene table that you can choose genes from. So this would be like a way to explore the output of EDGAR or DSeq2, for example. So Fiona worked on integrating the tree representation. So if you have a tree summarized experiment where you have, so this is like a summarized experiment where you can have a tree representation of either the rows or the columns. And with this kind of mode, you can visualize the tree, and you can also kind of select genes and or samples that you want to visualize, and you can show this kind of heat maps here of the expression levels of genes, for example, for the selected genes in a subset of the samples. Then what I was trying to make like some kind of proof of concept of was an idea from Laurent. So here you would have like a summarized experiment for proteins and an associated one for peptides. So they're individual objects, but they're linked in the sense that you know which proteins contain which peptides. So the idea here was to have two reduced dimension plots, one of the proteins and one of the peptides, and you want to kind of select a protein, and you would like to see where all the peptides of that protein are in the peptide PC. So that kind of was what I was looking into. Then we had Pierre-Luc, who was working on some kind of like GWIS or annotation representation, integrating EnsembleDB into an app. So here you kind of select a gene in a volcano plot, for example, and you would query the EnsembleDB package and get the gene model here in the other panel. And then finally we had Pablo, who was working more on like a, so it's not, I'm not sure if it will be called mode sneak peek, but it's kind of a first look of a dataset, single cell datasets, for example. You have your t-SNE plot, and you have your expression level plot of a given gene, and then you basically select a gene here in the table, a gene that you know about from before, a known marker gene, and so on, and you can see in the t-SNE plot which clusters express that gene, and also in the plot over here if it's expressed in certain tissues or in certain mice or something. So this is kind of what we were doing. I think we had a good time. I think we got a good bit on the way. If anyone else wants to contribute, that would be great. If you have a use case or something that you would like to have a mode for, we have you can join the IC Slack channel. Everything is in this repository. You can fork it and then make a pull request, for example. And I don't know if Kevin wants to say something about this, but IC 2.0 is coming with the next release, and there will be probably quite a big changes, at least in the usability. But I will stop here.
Yeah, before going to 2.0, I was just wondering actually for the users, what was the feedback in terms, let's say, of ease of implementation? Given the number of them, I assume that it was fairly easy. So people like picked it up and went along, or what's the typical time they take someone to get used to?
I think it went pretty smoothly. So I think most of the people had kind of seen it before, not necessarily used it. But I think it went relatively smoothly. So there are also already some examples, right? And you can like export the panel setting, and you have something to start from. So I think overall it went pretty smooth.
And one thing that's just crossing my mind is in terms of future panel, we probably need to think about discoverability. Sorry, that's a tough one. Of the panel, so that people don't necessarily re-implement the same panel multiple times, because at the moment, I guess it's only really the name of the mode that gives a hint about what it's doing.
Yeah.
So if I don't know exactly, I don't have a solution for that, but if people, let's say, have a concept in mind or some, I don't know, if we can ease the search of the panels, that would be useful.
Mm-hmm.
But yeah, I'm probably going to hand it over to Aaron to explain the refactoring, because that's literally his branch. I could give you the basics, but I think he's going to, it's easier if he just presents the concept.
Yes. Can you hear me?
Mm-hmm. Loud and clear.
Okay, good. All right. Okay, so can everyone see this screen? Where's the present? Okay, yeah, so I'll talk about version 2 that Kevin and I have been working on. So how this came about was during Thanksgiving, I had a vision, to make, to solve all of IC's problems, and this is 2.0. So the idea about 2.0 is that we're going to have a completely new architecture under the hood. Every panel type is now an S4 class, which, as I said, solves a lot of our problems. So we get natural validity checks on our input arguments. We get improved compartmentalization of the code, of the user interface elements, and of the observers. We have improved efficiency during app initialization, because basically only observers that are needed are set up, rather than setting up every observer for every possible panel ahead of time. And there's easier extensibility that I'll talk about later. Other cool features are that tables can now transmit selections based on the search parameters to the plots. So, for example, you can filter down a table to whatever you want, based on the columns of the table or the entries of the table. So you can filter that down and then transmit that selection to a plot, and that'll now work. Plots can now transmit single choices to other plots. So, for example, you can select a point in a plot, and then you can use that selection to indicate which gene should be used to color the points of another plot, for example. So that's also now possible. And there's our fun loading spinner. So when you're waiting for plots to load, you get this animation, which was pretty fun. Okay, so the S4 class hierarchy looks pretty complicated, but here it is. So the light gray boxes are the virtual classes, and the dark gray boxes are the concrete classes that you can actually call. Basically at each level of the hierarchy, there are different expectations as to what the concrete class needs to implement in order to support that concept. So for example, if you implement a column dot plot, then whatever concrete class you're implementing, each column of the summarized experiment object is expected to be a dot in some sort of ggplot. Conversely, for a row dot plot, every row of the summarized experiment is expected to be represented by a dot in a ggplot, and same for similar for the tables. If you're a column table, then each column of the summarized experiment should be a row in your table. The heat map doesn't really have any guarantees beyond being a panel. Right, so we have this hierarchy, and it's quite nice because it allows us to avoid repeating common pieces of code that are shared between, for example, the reduced dimension plot and the column data plot that we could just bundle into the column dot plot contract. So that was also quite nice. And internally we just do core next method, and then whatever whatever is in the column dot plot gets shared with whatever observers that we need to set up, or whatever interface elements that we need to define. We just core next method from a red dim plot into a column dot plot, and then we get everything that we needed.
So Aaron?
Yep.
Excuse me. Yeah, what's the difference between light gray and dark gray classes?
Oh, light gray is virtual.
Okay.
Dark gray is concrete.
Okay.
So this provides us with quite a bit of extensibility, because if you want to make a new panel, if you don't want to make a new panel class, then you can just inherit from an existing concrete class and specialize specific methods. So you don't even need to specialize anything if you don't want. but then there's sort of no point in making a new class. But you can make, like, on the right here we have something that Kevin set up, which is this reduced dimension hex bin plot, which is just the reduced dimension plot. But I think Kevin, you overwrote the plotting. You overwrote just the plot. Yeah. Okay. Yeah.
Yeah, it was mental how easy it was. It was literally just saying I'm extending the class of reduced dimension plot, and then there is just one function called getPlotting function, and I just hard coded ggplot hexbin with the right AES, and that was it, because the panel basically inherited the code to build the data frame that underlies the ggplot, because it's exactly the same as the reduced dimension plot. So all the work for the data wrangling was done for me. All I had to do is say I want a different different call to ggplot. So that part just requires having some convention that I'm sure Aaron can get in about telling users or panel developers what variables are available to them in the environment. So what can a ggplot call expect? And yeah, so at the moment we're working on the selection, which is getting a bit complex, but given the inheritance, it's really nice that the code is very dry in terms of specifying things once, having a nice generic and methods that are, if you override the methods at the right level, so sometimes you actually overwrite it at the virtual class level, and you can actually affect all the downstream panel because you want them to behave differently. It's a really powerful system in that way. It's a bit of a headache for us, so it takes me like 3 days of reading the code for 1 hour of writing something. But I think it's worth it for the end user, and in the future the panel developers will have a much better time writing new panels, because at the moment they just couldn't literally add new panels. The custom panels were very restricted in the features that they were offering.
So this is a good example of inheriting from an existing concrete class, and as Kevin said, you can also inherit, if something doesn't meet your needs, you just inherit from one of the virtual classes. So if you're making a new panel that where each dot is a point, then you can inherit from a column dot plot, and then write your own UI and observer elements. And if you're doing something even more weird, then you can just go all the way back to a panel object. And inherit from that. And each virtual class has certain guarantees on what it provides to the developer. So depending on what you want and what sort of how you want to interact with other panels in the app, you can choose the level in which you want to insert your own class. And the final really nice thing about it is that you can just add or override UI elements or observers for native integration into Shiny, which is something that we couldn't do with the previous custom panels. So there's quite a lot of power here in terms of how people want to make their own panels.
Aaron, have you considered composition for some of these aspects? I mean, I look at that class hierarchy, I can see, for example, row versus column as being an aspect that you might not want to bake into the inheritance hierarchy, but rather have, whatever concern, you have some separate module that implements row versus column, filtering or whatever the difference in behavior might be, and start to kind of split this out and compose these panels with different behaviors. So dot plot would be a behavior. How you're getting the data, or how you're indexing the data structure would be another aspect. You can kind of break that out. So I get a little bit worried about using inheritance for everything.
Yeah, I mean, actually, that is sort of happening anyway, because some of the aspects. So, for example, some of the aspects here, they actually have to use, like, some of the aspects in the dot plot class need to know whether it's a row or column. So they use a generic to indicate whether something is a row or column, and the generic is specialized for the actual concrete classes. So that sort of happens. That sort of is happening. I guess I just went with this because that's how I started.
Yeah,
that's how it is.
We always start with inheritance, and then we realize we went too far.
Yep. Yeah, well, that's how it is currently. It's all virtual, so it shouldn't be too difficult to change under the hood later on. Um, but yeah. Yep, that's me. Yep, I'm done.
Yeah. So
I
yeah,
had
no,
a,
I was just thinking. Yeah.
no, sorry.
No,
I
it's
had
go for it.
the linking. You had mentioned this linking behavior. I don't know if you're going to talk about that later or not, but I'm curious about how that's working. What is the mechanism you're using for the plot linking?
The linking behavior. Which one?
The link brushing or selection.
Oh, just the normal, right? I mean, we brush things, and then there's a selection of points that we transfer to another plot, and then we use that to do stuff.
It's a one-to-one because I mean you can imagine relational relationships right between different data sets where you might want to have some kind of custom function in there that determines the linking behavior. You might want to have multiple selections. You might have so I don't know, and you can even imagine maybe implementing as the column in a data set where you could even do things based on the selection. In other words, use it as a covariant in a model or something like that. I mean, you can imagine lots of ways of implementing linking. I'm just kind of wondering how
Yes. Okay. It's well, currently it's each panel has a single can receive from a single transmitter. So a panel can transmit to any number of other panels, but each panel can only receive. Or actually, sorry, that's not quite true. Each panel can receive one column selection and one row selection from two different. So like what it receives its row selection from can be different from what it receives its column selection from. So, for example, you could have a panel that recomputes PCA coordinates based on a selected subset of cells for a selected subset of genes. That's the most obvious use case. So that's in terms of the sort of structure of the links. And yeah, once you have those selections, then you could refit models with just your subset of columns or rows that you got from the other panels. Yeah, and that refit will update upon changes to those selections in the transmitting panels.
What about saving selections as a variable?
As a variable, so we have the ability to save multiple selections. So for example, you can select something and then you can save that, and then you can select something else and you can save that, and then you can so basically each panel can have a history of saved selections, and then all of those saved selections and the active selection can be transmitted to one of the receiving panels. So for example, you could use that to select, you have a t-SNE, you select one group of cells, then you select another group of cells, and then you select another group of cells, and then you can transmit all of those to another panel that does differential expression between those three groups of cells, for example.
How does that transmission work? What does that mean as a user? I mean, when I imagine selection, I see I select something in a plot, and then instantaneously another plot updates. What does it mean to transmit a selection?
Yeah, that's it.
But you're saving selections, and then those are also just appearing in the other plot. That's what's happening there.
What's saved is a brush. It's not the selection, right? You probably want to say that because you say each plot, actually you save the brush or the area of the plot when it's a lasso, but let's just keep it simple to say you save the brushes, one or multiple ones, and then that doesn't really do anything unless you tell another panel to listen to that panel with a selection. And so at that moment, the brushes are evaluated, and so there's a brush point function in Shiny. If you give it a data frame and of X and Y coordinates and a brush, it will just return you the subset of the data frame that's in that brush. So that's where we've extended the Shiny brush to be able to do that. So we store the Shiny brush, then we evaluate depending on whether the user wants the active brush, so the latest one of the saved one, or actually the union of everything. And so that's when the receiving panel, when it's time to decide what to show, will say, okay, I'm receiving the brushes from that other panel with the coordinates of the data points of that other panel. And then that's when we identify which of the points are selected. And depending on the user selection, what the user wants to do with the selection, we highlight them in a certain color, we restrict them in the plot. So that's when actually it's the ggplot call that just decides what to do. I know which points are selected. Now I want to color them in red, or I want to actually only show the layer of ggplot with those points extra. But the short answer is that the selection links in our case are just a list of what each panel is. Each panel has a parameter that says which plot do I receive my column selection from, my row selection from. If it's empty, nothing happens. If it's not empty and there is a selection in the other panel, then our code starts kicking in and adds the column to the data frame and adds the ggplot command to act on it.
Okay, that's interesting. Thanks.
Yeah. No worries. It's a bit of a long-winded explanation of it, because even the Shiny, the RStudio people, I think part of the prize we got was that we've implemented the lasso selection that they haven't implemented yet, and the multiple selection the way Aaron did it by just a saved history, which we added UI buttons. So it's literally we've pieced together Shiny features to make it work. But it is not a native Shiny feature to have multiple selection. That was one of the frustrations we had, or what people, I think users probably, a user request might have kicked that off. And we just said, okay, let's do it the way we can do it. But if Shiny comes up with a multiple selection feature, we might have to revisit that and say, because if they implement it, it's going to be native, it's going to be a lot easier to use, I expect.
Yeah, well, it's interesting because it's a persistent region selection rather than a persistent point selection. In other words, you have a brush, a rectangular or lasso-like region that the user has specified. And I guess if you were then to transform the underlying data and the points were to shift in the plot, then when you recompute the selection, the
it would be different. It would be different. I don't see. The points are not supposed to move, I think, in a plot. If it's a custom plot, okay, yes, I can see that. But all the sort of built-in plots,
if I may,
like reduced dimension,
if I may, if you change the nature of the plot in terms of the coordinates of the transmitting points, all the brushes and lassos are destroyed. So as to, well, I mean, they don't even make any sense if they weren't. So they just get wiped.
It seems very powerful, also somewhat complicated. I mean, having every plot dependent on a different set of selections from other plots. I don't know how it works in practice, but it just seems like, the simpler systems would just have one unified selection and all plots would be tied into that.
I mean, you could do that. I mean, you could just ask everyone to listen on a common pool. And that's something I thought about. But, I mean, it's actually no less complicated than the current system.
Okay. Internally or to the user?
Internally.
Cool. Thanks.
Cool. That was super interesting. Perhaps slightly more than a brief summary, but that's kind of what I was hoping for. That was really cool. I will try and give an actual brief summary of the Experiment Hub working group, because I don't think Laurent is here, and I've no idea what happened in the multi-assay experiment group. They wandered into our room at some point because they didn't have a projector adapter, and I'm not sure if anything happened beyond that point. But in our group, basically, we were a slightly bigger group. I guess there was maybe 12 to 15 people. Davide essentially gave a talk about Experiment Hub and the sort of principle behind why it exists, and how you might put data in there. And then we broke into 3 groups, which 2 were focused on essentially trying to prepare data for submission. So one was what I'm going to refer to as Elisabetta's data. She gave a talk on the first day about benchmarking single cell data sets. So you can probably find her slides and things. And Davide was essentially super excited about that data set and wanted to put it in Experiment Hub immediately. And then another group decided to take some of the 10x data that I guess is not there at the moment and try and learn how to package that up themselves. And then the group I was in kind of was looking at the documentation and perhaps like additional metadata and that kind of thing. So I don't know if either of the groups managed to get, the submission certainly didn't happen on the day we were there. I guess we always knew there was going to be you need to contact Laurie and get access to things. So there was always going to be a kind of roadblock on the actual day. One thing that we did find was that you cannot at the moment, I don't think, run the script that made the 10x brain data. HDF5 array has a problem when it tries to read the 10x matrix file and basically throws an error and won't proceed. I asked them to submit an issue, but I couldn't find it when I had a look earlier. So I'll see if I can recapitulate that problem and see if Hervé and I can work out what's going on there. And then, the reason that the group I was with looked at the documentation is that essentially, I guess what's written in the vignette doesn't really marry up, certainly with the discussion we had on this teleconference a couple of months ago about maybe the best strategy is to break the data up into the component parts and then have some sort of building mechanism that recapitulates a single cell experiment or something. I think the documentation sort of says we expect to have complete objects and that kind of thing in there. So we were basically looking at maybe we could make some adjustments to that, although we haven't also submitted any pull requests or anything like that. And then also whether we could add additional metadata like the paper that a data set has been published in and that kind of thing to the store in Experiment Hub. So we learned quite a lot about the hierarchy of the hubs and the hub data packages and how that's all structured and things, and realized this was going to be way more work than 2 hours on a Wednesday afternoon. So I think probably I need to contact Laurie and see whether these kinds of things are useful for us to continue investing our time. But it was a fun afternoon in our little group, and I guess I'll probably leave it at that. So we had far less concrete results than the IC guys did.
Yeah, like if you want to schedule a chat at some point, we can review all that stuff and see what would be worth pursuing. And obviously, feedback is always welcome to documentation and everything.
I'll mention that one thing that Kayla has been working on, I mean, if she wanted, but so the way that the contributing data to the hubs, especially experiment data, is centered around a package, and Kayla has written a kind of a helper function that creates the Experiment Hub data package or a hub data package to sort of facilitate that kind of the way dev tools create. I think that's on a branch, but not yet merged.
I think one thing we realized was that a lot of the data packages have kind of the same code reproduced over and over again to build a single cell experiment or something from the component parts, and basically each package implements it themselves. And so maybe actually putting that either in another package or in Experiment Hub itself or something might be a more sort of centralized place for one tool rather than everybody reinventing how to do this each time.
I don't think we originally, because again, now that the data is larger, we originally recommended it being a constructed object rather a deconstructed object. But I believe that there's, it's implemented in at least Annotation Hub where you can have like the linked files, like the BAM and BAM index files, where maybe it's worth pursuing looking into seeing If that's still supported, I have a feeling it is in Experiment Hub, and then it would be creating like a new dispatch class with the related files and having like a new dispatch class for single cell experiment that would do the construction for you. But I would have to investigate that a little bit more.
Yeah, this was very much sort of pie in the sky thinking about what might be nice, but without any concrete plans for how you might go about doing it. So if we've got nothing more to cover about the EuroBIOC stuff, I will just share my slides, my screen with some slides about HDF5. Is that showing up for people?
Yep.
Cool, excellent. Yeah, so this is basically just some work which I guess I actually started about 2 years ago and then stalled and then came back to it again a couple of months ago, which is basically about trying to see if there are other compression tools for writing data to disk that might be more effective for the kind of data that we work with, and how we could distribute that to people in a relatively transparent way. So I guess most people on the call probably know about HDF5 files and things, but there's a couple of slide summary, right? The data sets themselves might be large and typically in our single cell world kind of rectangular, but they are not actually stored as one big contiguous thing, right? They're stored as chunks, and those chunks on disk are actually separate from one another. And so in our little picture here, we're just representing 5 by 5 kind of chunks of data. And if you want to read a subset of the data, for instance, just one point, right, you only need to read the chunk from disk that point appears in. And we can choose the shape of the chunks and things to try and optimize this if you know about the data access pattern and that kind of thing. But that's really the kind of power of HDF5 is this kind of chunking and relatively quick random access into the file. One thing that happens with those chunks, right, is that they get processed by filters. So when you're going from memory and writing them into disk, this slide shows the kind of standard HDF5 pipeline. So each chunk of data goes through a shuffle, which basically reorders the bytes into an order that's hopefully more easily compressible, and then they go into the standard gzip zlib compression algorithm and then get written to disk. And then when we read them from disk, the filter pipeline gets put into reverse. So it decompresses them, then it unshuffles them, and then you get them back into memory again. And this filter pipeline is kind of flexible, so you could take the shuffle bits out. You could change the compression algorithm in theory for another one, or chain two together if you know that happens to work more successfully or anything like that. So it's quite a flexible pipeline. You have to be able to do whatever you do in one direction, you have to be able to undo it in the other direction. And so those filters and the tools for doing them basically have to be available on whatever computer is trying to do. If you give some data that's been written to disk, whoever's reading it needs to have the same compression algorithms available on their machine to be able to work with that data. And so sometimes people get errors where if it hasn't compiled properly, if they don't have zlib on their machine or something, they'll get some standard HDF5 data and get a weird cryptic error saying it doesn't, you're unable to read it. And so there's a number of compression filters available, but basically HDF5 itself and our HDF5, our version that we ship with Bioconductor, they both come with support for gzip, so the kind of standard compression tool that you get in zip files and things, and then also something called szip, which is, I think, pretty much bespoke to HDF5 is developed for some sort of NASA satellite imagery kind of thing, and I've never really read about it in any context other than HDF5 files. So those ship with the source, and if your data is written with either of those, then it should be fine to read it. And almost every tool, whether it's R or Python or the C library or whatever, is probably built with support for those. But then there are what they call dynamic filters, so they don't ship with the source, but basically they're third-party tools. So if you take a third-party compression tool, you can wrap it in a little bit of extra code that allows it to talk to our HDF5, basically. And then you can compile those into shared libraries, and if you provide the location to HDF5 via an environment variable, they just get loaded when they're required. So if you specify that they're needed for writing, it will load them. If they encounter a file that has been encoded with one of those, it will look in this location, and if the decoder is there, it will use it on the fly. And because they're kind of compiled independent of whatever you're using, you can use R to write something, and then if the filter is available, somebody with a Python application could read it, or vice versa, that kind of thing. So this is kind of a flexible way of being able to maybe distribute these sorts of things without having to recompile your software every time somebody updates the filter, or you get a new data set or something like that. And so I've bundled a couple of these into an R package. So there's bzip2, which is, I guess, quite popular. BZ2 files are found all over the place. It tends to be pretty good at compressing things, but takes a long time. And then also the BLOSK meta compressor, which is an effort to build a wrapper around 5 or 6 other compression tools. So there's kind of several layers of tools helping each other out here. But I bundle both of these up in this kind of dynamic filter format, and basically ship that C code. So the libraries plus the code that turns them into filters shipped in a package, and then that gets built or compiled when you build the package. And I managed to get that working on on all the platforms, including Windows, which is where I failed 2 years ago to actually manage to get a useful distribution of this. And then I've been doing some work to integrate it with our HDF5 itself. So basically, if you use the kind of high level, I want to write an HDF5 file kind of approach, you can basically now just supply an argument and say, I want to use bzip2 or BLOSK or whatever, in the same way that you would set that I wanted to either turn on or turn off the standard zlib compression previously. And then also, if you get given a file and try to read it with our HDF5 and it's used one of these, it will just use them automatically. So at the moment, it's not in Bioconductor, it's just on GitHub and things. But in my tests, it seems to have been working reasonably well so far. So to try and figure out how well it's been working and whether it's any particular settings are more appropriate for our data and things, I've been trying to benchmark a few things. So some single cell data sets and some randomly generated data and things, and then just comprehensively running through all of the algorithms on all of their different compression settings and things, and then just measuring how long it takes to read things, how long it takes to write them, and how big the files that come out are. And if you're interested, like there is no clear winner in terms of it depends what you want to do, how many times you read it versus how many times you write it, and that kind of thing. But there's clearly big differences between some of the approaches. And if you're interested in exploring that data, there's a Shiny app. So I guess I've actually got it open somewhere. This one, yeah. So there's this is running locally, but there's a GitHub repo where you can download it and run it yourself. And basically, this bundles up all of my benchmarking runs, and you can choose to change some of the, look at how it changes with different parameters and that kind of thing. So you can look at which algorithms there are, how it's structured, how it's ordered, and the different benchmarks that I've run. So it's just a way of trying to give people access to look at these benchmarks, so we can make some informed decisions about whether it's useful or not. So I think that's basically the presentation. Happy to take any questions.
Mike, do you think you could use this filter, this compression mechanism for encoding sparse data using conventional sparse matrix representations? And just kind of do it transparently that way?
I don't know is the honest answer. Essentially, it will. I guess it just treats each chunk as a stream of bytes, and you can do whatever you want with data. So the shuffle thing that is built into it will just move the bytes around, but it doesn't actually compress things at all. So, but you could make the file bigger if you really wanted to, I guess, by multiplying everything, or which would be a crazy thing to do. But essentially, you can do whatever you want in these filter steps.
Okay. So essentially, it allows you to create your own chunk storage format.
Exactly. Yes. I think the filters are kind of agnostic as to what they're actually doing, right? Like almost all of them are for compression, but they don't really have to be.
Cool.
Right. You could use your own RLE compression, for example, by your own RLE filter.
Yeah, I think so. Yeah.
And you can see the sparse representation as one way of compressing the data. So that, in theory, you should be able to do that too.
Yeah, I think there's no real limit on what we could do with it. As long as we can write the code, we could do whatever we want, basically. I've picked existing compression tools, I guess, and just written the kind of simple wrappers around those. But there's no reason why we couldn't write the compression tool as well, or reuse other code that isn't that I haven't looked at so far.
I guess there'd be no way to make it, for the way I guess the HDF5 is architected, when it queries the data, random access, it first figures out which chunk you're in, and then it just, I guess, streams over the compressed data, decompressing it until it finds that element. Presumably that's how it works, just like the other, blockwise compressed file formats. If that's the case, I guess because like one thing you might think about would be, could we optimize access by making somehow a randomly accessible block? So in other words, like you could figure out some compression algorithm that would allow you to also perform random access, like maybe using one of these simpler sparse matrix representations. It's not like low-level compression. You might be able to figure out where in the block a particular value is without having to stream over the data.
Yeah, so I don't actually know how it does, whether you can jump into the middle of a block. I've no idea whether that's possible or not, or whether it will default to
anyway. Won't let you do that because it's probably designed to just stream over the compressed data through this filter. So there's no way to do it. But if we went under the hood, we could potentially do it faster.
Mike, so this integration, so this integration of RHDF5, I assume happens at the R level, as in terms of if it sees a filter it doesn't recognize it, then sucks in RHDF5 filter to provide the
So at the moment it's a bit. I think this is one of the questions I have. It's in some way where should it? Which package should depend on which one, right? Because I think in principle you could just use RHDF5 filters as it just distributes the shared objects or the code to build them and has no R code in it at all, except for when you load the package, it sets one environment variable, and that's basically the location that everything should look in after that. Which is kind of how it's set up at the moment. There is a bit of R code which links back to RHDF5, and that gets me stuck in this kind of dependency cycle. And I don't think that's the right way of doing things.
Right, because
Sorry.
Because the problem I'm thinking of is for the various packages that I have that depend on rhdf5 lib. Right. So what happens if so those packages also depend on rhdf5, but they don't access the hdf5 file via rhdf5. They access it via the C level hdf5 library. And then I guess it'll just crap out, right? If it can't find it won't know where these filters are.
So I guess I could make the filters package be loaded when rhdf5 lib is loaded, so they get loaded at the same time.
Yeah,
because and then sorry.
If we want this to get a format for exchanging data, people have to be able to access this outside of R even. So if we're implementing our own filters, they need to be done at the HDF5 library level, system level.
So yeah, I guess this is why everybody just uses the zlib compression, because it ships by default, and that means you've got a pretty good chance that if you move the data to somebody else, whatever tool they're using. But there's certainly the filters that I've taken were initially being, well, my efforts are based around things from the Pi tables function or suite of tools and things. So there are other people that have definitely implemented the ability to read rhdf5 files with BLOSK and with bzip2. And I know a couple of other people have contacted me asking if you can read things compressed with certain tools in rhdf5, and my answer has always been no at the moment. So I guess maybe we test this and we decide that the lack of portability is not a great idea. And that's fine. Then I've done this and we've tested it and we decide that it's not worth the benefit. But at the moment, I guess I'm still keen to explore and sort of create the opportunity to be able to use these other things.
So Mike, so if we think in terms of the C level, if we think in terms of the C level, how is it finding these filters again? Just technically
it
speaking.
just sets an environment variable.
Okay.
Called like HDF5 plugin library or something like that. And so I've used the version that I built from rhdf5 filters, so built as an R package. And I give that environment variable to the like HDF5 GUI viewer thing for reading data sets. And that works. And that's like completely independent of R.
Right. I think the question is, an important question is which filter was used, recorded, and in the file itself. The file needs to be self-descriptive.
It's stored in the data set, the data set metadata within the file.
What is
it's
stored there?
Just an integer value. So some of these are registered with the HDF5 group. So
I see.
I guess they have a specific name and nobody else should use that registered. Like there's a page with a list of maybe 25 filters or something, a lot of which when you try and find the code base, it's dead, and they're very specific for certain image formats from one research group somewhere and things. But yeah, essentially there's this integer value that represents the compression tool.
I see.
And knows what to use when. So it just looks and says, have I got a filter available to me that has that same ID value? And if it doesn't find one, it will throw an error. Well, by default, a very long and complicated error, but every error in HDF5 is a long and complicated error.
Right. So yeah, basically, it's the whole scheme assumes that the number, there's a lookup mechanism that is reliable and a central authority for maintaining that lookup.
Yes. Yeah.
Yeah.
It seems like if we want to use this from the C level, we'll need another abstraction layer on top of RHDF5 lib that mediates the use of the correct filters from RHDF5 filter when it encounters when we try to open a file, basically at the C level. And then I just call that particular abstraction layer, and then the fact that the developer would never need to care where these filters come from, as long as they were one of the filters supported by RHDF5 filter. Because currently, if we wanted to use the current situation, then if I get a file that is not part of the standard filters, I then have to go and hunt down. I have to write the code to pull in the RHDF5 filter shared library in order to use that.
Yes.
To get it to work.
Yeah. You need to have built the shared library somewhere and know where that location is, and then provide it as a environment variable, and then your C application will be able to read it.
So for example, one way to do that, if we had some way, if HDF5 provides some interface for pulling out the filter, beyond a try and catch. If it has some way of pulling out the filter identity, then you could check whether it's one of the ones supported by RHDF5 filter, and then pull in the shared library. That's just one way of doing it, right? But you could imagine doing it like that.
Yeah, so you can access the filter number with it's in the dataset creation property list. I think that's the right term for it. So when you create a new dataset, you set the filter value. So most of the time we just set it to whatever the value for zlib is. But and then you can always read what the creation properties were on an existing file. And this is just one of the values that's stored in there.
I see. Our filtered version is a mechanism to store a filter version associated to the filter itself. I can see. I'm just starting to worry about this. It's too early, but you can imagine that the filter has changed because the bug was found in the filter. So now there's a new implementation for that filter, and HDF5 files that were created with the previous version of the filter now are not backward compatible with the new one. You can imagine all kinds of things, right? If the way the filters are recorded in the file is not precise enough. So I was wondering about the versioning.
If there's filter versioning. I can't remember whether there is or not. But I guess there are many ways that you can make HDF5 files that are incompatible with older versions 5. Like there was quite a long time where we shipped 1.8, and I guess it was Callisto was making things with 1.10, and nobody could read the files that it was creating. That was one of the motivations for updating our HDF5 lib distribution. And in theory, you could make them backwards compatible, but they weren't being made with that setting turned on. I guess there are many options and many ways of making HDF5 files not work, but and we should probably pay attention to them. But I guess you can't mitigate every possible circumstance.
Yeah.
Cool. In which case, we've used our hour. So thank you very much. Does anybody else have anything they want to add, contribute? Great. In which case, I wish everybody a Merry Christmas and see everybody in the new year.
Yep. See you guys.
Bye.
Bye. Happy Christmas.
Bye.